IB Bio, Cell Respiration Part 2, has its focus on the first series of reactions in cell respiration, and the first part of cell respiration is called glycolysis. Most of Part 2 will be for higher level students, but glycolysis leads us right to anaerobic respiration, also known as fermentation, and SL students are responsible for this material. SL students, you need to be patient working through some HL material to get to SL material. The IB syllabus is not constructed in ways amenable to clean lines between the HL and SL material in this unit. The essential ideas are cell respiration supplies energy for the functions of life, and energy is converted to a usable form in cell respiration. Here is the outline of all the movies available for the cell respiration unit. Use this outline to find the movie you need for review. This movie is focused here, covering glycolysis, fermentation, and this movie introduces the link reaction, which is covered again in Part 3. Cell respiration is central to the metabolism of a cell. This diagram displays hundreds of metabolic pathways that occur in a cell, and this is just a fraction of all that occur. You can see the outline of the mitochondrion here. The role of the mitochondrion as an organelle that transforms energy is vital to life. Cell respiration happens mostly in the mitochondrion within a cell, but glycolysis, the subject of this movie, occurs in the cytoplasm. Respiration is the controlled release of energy from glucose to drive ATP synthesis. ATP leaves the mitochondrion and is hydrolyzed, broken down, in the cytoplasm to do cell work, such as active transport or movement. Then ADP and the inorganic phosphate are covalently joined to form ATP by the energy released during cell respiration. Here are the first two IB syllabus statements of the movie. In glycolysis, glucose is converted to pyruvate in the cytoplasm. State that glycolysis gives a small net yield of ATP without the use of oxygen. Glycolysis, seen here as the first set of reactions in cell respiration, occurs in the cytoplasm. Some ATP is produced as a product, and in glycolysis, glucose is converted to pyruvate. Now notice one other aspect of this diagram. Electrons are removed from the organic intermediates of glycolysis and carried to the electron transport system of the mitochondrion by a molecule known as NADH. NADH is an electron carrier, and we'll understand the significance of NADH and a relative of NADH known as FADH2 when we look at cell respiration, Part 4. This diagram provides more detail on the series of reactions in glycolysis. This diagram remains quite simplified, as not all the reactions are shown. In glycolysis, glucose, starting with 686 kcals per mole of free energy, is energized with the expenditure of 2 ATP molecules. The upward direction of these two steps is indicative of the added energy to glucose. Then, the intermediate organic molecules are oxidized to give up energy, and 4 ATP molecules are synthesized, 2 here and 2 here. 2 spent, 1, 2, and 4 synthesized, 1, 2, 3, 4. There's a net gain of 2 ATPs in glycolysis. Pyruvate, or otherwise known as pyruvic acid, is the end product, having less free energy at 543 kcals per mole. Let's dig a bit deeper with 3 more IB syllabus statements. Describe glycolysis as a metabolic pathway in the cell cytoplasm. List 3 products of glycolysis. During glycolysis, identify the following events: phosphorylation, lysis, oxidation and reduction, and ATP formation. Glycolysis is a metabolic pathway in the cytoplasm. All of these reactions occur in the cytoplasm, and pyruvate, also known as pyruvic acid, is also formed in the cytoplasm. The products formed during glycolysis are pyruvate, ATP, and NADH. But let me take you through glycolysis more carefully. Listen for the following events: phosphorylation, lysis, oxidation reduction, and ATP formation. The free energy of glucose is increased when glucose is phosphorylated by the expenditure of 2 ATPs. The addition of phosphate groups makes this molecule, fructose 1,6-bisphosphate, unstable and more ready to enter the next series of reactions. Fructose 1,6-bisphosphate is cut into 2 3-carbon molecules. This step is called the lysis step because the 6-carbon molecule is lysed into 2 3-carbon molecules. This is important because from now on, we must follow 2 molecules through every step in order to account for everything. Following lysis, the 2 organic intermediates are then oxidized as electrons and hydrogen ions are removed. In turn, NAD+ is reduced to become NADH. This is your oxidation reduction step. 2 NADH molecules are formed because we have 2 intermediates proceeding through this reaction. Another phosphate is added to each intermediate in a non-energetic step, and then each organic intermediate gives up a phosphate to ADP to form ATP, not just once, but twice. 2 ATPs here and 2 here. 4 ATPs total are formed as 4 ADP molecules are phosphorylated. The end product is pyruvic acid, also known as pyruvate. There are 2 pyruvate molecules here because of the lysis step back here. Also notice that all 6 carbon atoms of glucose are still present. In other words, no carbon dioxide has yet to be removed. Here is the lysis step. Fructose 1,6-bisphosphate is lysed into 2 molecules, each of which proceeds through every metabolic step until all the carbon has been removed as carbon dioxide. By the way, we can see the expenditure of 2 ATPs to energize glucose, forming the more unstable fructose 1,6-bisphosphate, unstable and ready to be lysed in the next reaction. We can also see the formation of 4 ATPs here so that we have a net gain of 2 ATP molecules. 2 are spent and 4 are formed, a net gain of 2. The next syllabus statements are: state that the phosphorylation of molecules makes them less stable. When glucose was phosphorylated to fructose 1,6-bisphosphate, the molecule was less stable and more readily entered the lysis step, where it was broken down into 2 3-carbon molecules. The second IB syllabus statement is: describe the role of reduced forms of NAD and FAD in carrying energy following oxidation reactions. Now, we've not seen FAD yet, but NAD+ grabbed electrons and hydrogen ions from the organic intermediates of glycolysis to become reduced as NADH. Reduction is gain of electrons and/or hydrogens. Now, NADH carries electrons held in high energy shell positions surrounding an atomic nucleus. NADH carries electrons to the electron transport system of the mitochondrion. The phosphorylation of glucose, or the phosphorylation of ADP, makes the molecule ATP more unstable and more readily available to enter a reaction. This is what makes ATP such a good energy molecule. Now, NAD+ takes electrons and hydrogen ions from the organic intermediates of glycolysis. The intermediates are oxidized as they lose electrons and hydrogens. The NAD+ is reduced as it gains electrons and hydrogens. And here are the relevant IB syllabus statements. Define oxidation reduction reactions and describe the role of coenzymes, NAD and FAD, in redox reactions. Cell respiration involves the oxidation and reduction of electron carriers. Analyze diagrams of the pathways of aerobic respiration to deduce where decarboxylation and oxidation reactions occur. Oxidation is the loss of electrons or hydrogens. Reduction is the gain of electrons or hydrogens. In this diagram, methane is oxidized, losing electrons and hydrogens to oxygen, while the oxygen is reduced as it gains electrons and hydrogens. Highly electronegative atoms, such as oxygen, draw electrons towards its nucleus with the release of energy. I covered a bit of the details of this in Part 1. Go back to review Part 1 if none of this seems familiar. NAD plus and a relative we've not yet met, FAD, serve to oxidize organic intermediates. NAD plus takes electrons and hydrogens from those organic intermediates and becomes reduced to NADH. NADH carries those electrons held in high energy positions around atomic nuclei to the electron transport system of the mitochondrion. As we move through all the stages of cell respiration, oxidation reduction will feel more and more comfortable. Once again, glucose, a 6-carbon molecule, is phosphorylated twice, becoming a more energetic, unstable intermediate molecule that is lysed into 2 3-carbon molecules that are oxidized by NAD+, which is in turn reduced. Phosphate groups are removed from the intermediates in the formation of ATP. This happens twice to each of the 2 molecules for the production of 4 ATPs. 2 molecules of pyruvate remain at the end of glycolysis. The energy loss from glucose to pyruvate can be accounted for in the ATP, NADH, and some heat. Having memorized the summary equation for cell respiration, it should have crossed your mind that we have not yet seen oxygen as a reactant or carbon dioxide as a product. So far, the part of respiration we've studied has been anaerobic, no oxygen required. So here is the relevant IB syllabus statement that states what we already know. Anaerobic cell respiration gives a small yield of ATP from glucose. And that takes us to the next detail, something that we need to study. Explain that in anaerobic cell respiration, pyruvate is converted into lactate or ethanol and carbon dioxide in the cytoplasm. When oxygen is present during aerobic respiration, the end product of glycolysis, pyruvate, moves to the mitochondrion along these pathways. But without oxygen, anaerobic conditions, pyruvate is fermented to lactate, lactic acid in mammals, or ethanol in yeasts. You can see that lactic acid is a 3-carbon molecule, just like pyruvate, but ethanol is a 2-carbon molecule as carbon dioxide is removed in the step from lactic acid to ethanol. Fermentation, as seen here, uses the reducing power of NADH to transform pyruvate into lactate, returning NAD+ to glycolysis. This allows glycolysis to continue in the production of 2 ATPs. While glycolysis does not produce a lot of ATP, it does produce some. And without NAD+ to oxidize the intermediates of glycolysis, glycolysis would come to a halt. NAD+ concentrations are maintained even when oxygen is absent because NADH is oxidized as it reduces pyruvate. Thus, NAD+ returns to the glycolytic pathways to oxidize the organic intermediates. This is all in an effort to maintain small quantities of ATP production. Glucose to pyruvate is glycolysis. 2 ATPs are produced. In mammals with no oxygen present, pyruvate is fermented to lactate. Notice how fermentation allows NAD plus to be regenerated to make it available for the oxidation of the organic intermediates in glycolysis. And this is for the continued production of small amounts of ATP. Glucose to pyruvate is glycolysis. 2 ATPs are produced. In yeasts, when no oxygen is present, pyruvate is fermented to ethanol with the loss of carbon dioxide. Notice how anaerobic respiration, fermentation, allows NAD plus to be regenerated to make it available for the oxidation of organic intermediates in glycolysis for the continued production of small amounts of ATP. In yeasts, when oxygen is not present, pyruvate is fermented to ethanol with the loss of carbon dioxide. This loss of carbon dioxide is called decarboxylation. Notice how the NADH is oxidized as the organic intermediate is reduced. This allows NAD plus to be regenerated to make it available for the oxidation of organic intermediate molecules in glycolysis. The organic intermediate is oxidized as NAD plus is reduced to NADH. This is an oxidation reduction step, and this is an oxidation reduction step, all in an effort to maintain a small amount of ATP even when oxygen is not present. Remember, there is no loss of carbon dioxide from pyruvate when pyruvate is anaerobically fermented to lactate in mammals. NADH is oxidized as the pyruvate is reduced. NAD plus is made available to glycolysis. This is an oxidation reduction step, and this is an oxidation reduction step. At this point, you should feel comfortable with this syllabus statement. Explain that in anaerobic cell respiration, pyruvate is converted into lactate, or ethanol and carbon dioxide in the cytoplasm, with no further yield of ATP. But let's review a bit of detail. In this diagram, you can see that glycolysis is located in the cytoplasm as glucose is converted to pyruvate. If oxygen is present, then pyruvate is moved into the mitochondrion. If oxygen is not present, pyruvate is anaerobically fermented to ethanol or lactate as a mechanism to maintain concentrations of NAD plus in the cytoplasm. Glycolysis and the production of small quantities of ATP require NAD plus as a means to oxidize organic intermediates in glycolysis. If oxygen is present, pyruvate is moved into the mitochondrion to continue aerobic respiration. If there is no oxygen present, pyruvate is fermented into ethanol in yeasts and lactate in mammals. Electrons and hydrogen ions are pulled off organic molecules to reduce NAD plus to NADH. NADH transports the electrons held in high energy positions to the electron transport system. There's a lot of free energy in the electrons held by NADH, and those electrons will be delivered to the electron transport system. NADH is called a coenzyme, and NADH contains stored energy. NAD plus is an oxidizing agent as it oxidizes the organic intermediates in glycolysis, seen here. NAD plus grabs electrons and hydrogens to become reduced, forming NADH. NADH shuttles electrons held in high energy positions to the electron transport system of the mitochondrion. This is an oxidation reduction step. As a note, a side note, the enzyme that catalyzes the formation of fructose 1,6-bisphosphate is allosterically inhibited by ATP. Let's take a look on the next slide. When a lot of ATP has been produced during cell respiration, it would be beneficial to a cell to stop producing more. As ATP concentrations increase, ATP acts as a non-competitive inhibitor on PFK, phosphofructokinase, which is the enzyme catalyzing the formation of fructose 1,6-bisphosphate. When oxygen is available to the cell, pyruvate moves into the mitochondrion. Remember, pyruvate still contains substantial energy per mole, and that there are 2 molecules of pyruvate formed for each molecule of glucose that started glycolysis. 2 molecules of pyruvate, each with 3 carbons, are products at the end of glycolysis. Note that no carbon dioxide has yet to be released as waste up to the point of pyruvate formation, but that's about to change as pyruvate enters the mitochondrion. Under aerobic conditions, pyruvate enters the mitochondrion. The link reaction follows. But before looking at the link reaction in detail, let's use this slide to organize ourselves. The 4 sets of reactions in cell respiration are glycolysis, the link reaction, the Krebs cycle, and electron transport system. This movie has covered glycolysis and is about to introduce the link reaction. Part 3 looks at the Krebs cycle, and Part 4 looks at the electron transport system. Here's the relevant IB syllabus statement. Describe how pyruvate is decarboxylated and oxidized and converted into an acetyl compound and attached to coenzyme A to form acetyl coenzyme A in the link reaction. The series of steps as pyruvate enters the mitochondrion is called the link reaction. Pyruvate is transported into the mitochondrion, and immediately carbon dioxide is removed in a decarboxylation step. NAD oxidizes the 2-carbon intermediate known as an acetyl compound, and the acetyl compound is immediately attached to another coenzyme known as coenzyme A, also known as CoA. The acetyl compound, along with coenzyme A, is called acetyl-CoA. Acetyl-CoA enters the Krebs cycle. The link reaction results in the following products: 2 carbon dioxide molecules, 1 for each pyruvate, 2 NADH molecules, 1 for each acetyl compound, of which there are 2, and 2 acetyl-CoA molecules ready to enter the Krebs cycle. And now, these syllabus statements should feel familiar. Define oxidation-reduction reactions and describe the role of coenzymes NAD and FAD in redox reactions. Cell respiration involves the oxidation and reduction of electron carriers. Analyze diagrams of the pathways of aerobic respiration to deduce where decarboxylation and oxidation reactions occur. So returning to this diagram, here is decarboxylation, here is oxidation reduction. The acetyl compound is oxidized while the NAD plus is reduced to NADH. NADH is a coenzyme that carries electrons held in energetic positions to the electron transport system. Here is a diagram of a cell. You can see glycolysis having happened in the cytoplasm to produce pyruvate. Pyruvate enters the mitochondrion and the link reaction occurs. You can see decarboxylation here and oxidation reduction here, and the 2-carbon acetyl group added to coenzyme A enters the Krebs cycle. The Krebs cycle will be the subject of cell respiration, Part 3. This is a diagram of the mitochondrion. Glycolysis happens in the cytoplasm. If oxygen is present, then pyruvate is shuttled into the mitochondrion, as can be seen here. The next 2 stages of cell respiration, the link reaction and the Krebs cycle, happen in the matrix of the mitochondrion. You can see NADH molecules containing free energy transporting electrons to the electron transport system. Here is a table that accounts for everything. You can see that glycolysis produces 2 moles of NADH per mole of glucose, 2 moles of ATP net per mole of glucose, and 2 moles of pyruvate, which could have different fates depending on the presence of oxygen or not. If oxygen is present, Then pyruvate enters the mitochondrion, initiating the link reaction. The link reaction results in 2 moles of carbon dioxide for each mole of glucose, and 2 moles of NADH per glucose, and 2 moles of acetyl-CoA ready to enter the Krebs cycle. One last detail before leaving this movie, a detail that's important in mentioning before going forward, is that ATP formation through glycolysis and the Krebs cycle is different than it is in the electron transport system. In glycolysis and the Krebs cycle, ATP is formed by a process known as substrate-level phosphorylation. A substrate molecule with an attached phosphate group gives up the phosphate group to ADP, as you can see here. In this case, PEP is the organic intermediate, the substrate, and an enzyme catalyzes the transfer of the phosphate group to the ADP to form ATP. Now, this is not a huge issue, but substrate-level phosphorylation is not the same process that we will see later in the electron transport system, and I just wanted to make the distinction. Here's one last image of substrate-level phosphorylation. An enzyme transfers the phosphate group from a substrate to ADP to form ATP. You can see that substrate-level phosphorylation is the type of ATP production in glycolysis and the Krebs cycle, but not in the electron transport system, where it's called oxidative phosphorylation. And that brings us to the end of IB Bio Cell Respiration Part 2.